We're going to welcome up Michael Peluso with the introductory overview of HIV cure research from University of California, San Francisco. Everybody welcome Michael Peluso.
This is Michael Peluso.
Hey, everybody. I apologize for my height. I'm not tall enough for this podium, so I'm going to stand on my toes. Yeah, I'm Michael Peluso. I'm a HIV clinician and researcher at UCSF, working on our cure program, and it is such a pleasure and a privilege to be here. This is actually my first time here today. So I'm going to try to provide a broad overview of HIV cure in 2025 and beyond. And I'm going to ask Michael Luella, who is advancing the slides, to do so. So I'm going to begin by talking about where I think we are, and then I'll go on to where we need to go and how we're going to get there. So where we are. Next slide, please. So, in 2025, ART is pretty great. It is terrific for prevention. It is terrific for cure. When I talk to my patients in clinic about, my role as an HIV cure researcher, they often ask me, well, do you work on long-acting ART? Because for many people, the reality is that, long-acting ART with infrequent dosing of injectable medicines is approximating a cure, or at least what some people think a cure could look like. And so, I think it's important to talk about what we're trying to accomplish in the context of the massive improvements in ART that have happened over the last few years. Next slide. So I'm okay. I don't know if they can hide that bar. I'm getting. So why do we need to be talking about a cure in an era of highly effective ART? So everybody in this room knows these reasons, but there are human costs to the current need for ongoing treatment. This includes both sort of external discrimination and stigma, as well as internal stigma from the act of taking medicines every day. We know that there remain long-term adverse health outcomes related to living with HIV, including many comorbidities that are more common among people living with HIV, as well as issues related to polypharmacy, so having to take multiple medications, especially as we get older. Some people have resistant virus and aren't really eligible for one pill once a day or the injectable medications. And even for the most adherent people, lifelong adherence is really challenging. I mean, just filling meds at a pharmacy is like way more complicated than it needs to be. And certainly on a global scale, this remains a big challenge. And then, even thinking back before 2025, despite all of the investments that we have made in operationalizing ART, too many people are not able to access it and remain on it. So those are big challenges. Next. And then I think, for many years, we've been talking about the public health challenges. And I think, unfortunately, in the last 6 weeks, we have seen many of these challenges shift from being kind of more theoretical challenges that we worry about to like very, very real challenges. We saw this over the last 5 years with COVID causing this huge disruption in people being able to access meds. But now we've seen this sort of ramped up, unfortunately, even more with disruptions to programs like PEPFAR and USAID, which we may not necessarily think about as cure programs, but everything is so highly intertwined that, the challenges that these programs are facing now are going to affect all of us, regardless of our focus within the field. Next slide. like the real-time emojis here. So much of the progress in the last, I'm gonna just stand here. So much of the progress in the last 10 or 20 years has really been in getting a deep understanding of the challenges that we need to overcome in moving toward an HIV cure. And I'm not going to spend a lot of time on this, but there are challenges, related to the reservoir, understanding how and why and where HIV is able to persist. And we have a better understanding of that now. We had hoped that over time we would see the reservoir sort of, decline just continuously in people. We have seen that that does not really happen on ART. There's an initial decline, but then it sort of levels off. And in some cases, it may even go up over time because of issues like clonal expansion. And so there's a lot of heterogeneity in how the reservoir changes. It's a really tough challenge to overcome. We know that even small amounts of virus deep in the tissues can reemerge when people pause their meds. And so we can see, on all these advanced PET scans and stuff, single lymph nodes basically lighting up with reactivation when people are off of meds. We're coming to understand all of the different arms of the immune system that are really important in sort of helping to control HIV in a person's body and potentially that need to be further honed to cure people of HIV. Next slide. So as everyone here knows, there are at least 7 people who have been truly cured of HIV by stem cell transplants. And I think that this is one of the most, inspiring things that we have observed as a researcher. I think that this provides a tremendous amount of hope that this is possible. But I mean, honestly, we all know that this is not scalable, right? So the importance of these cases is huge, but this is not going to be the answer for everyone. Next slide, please. I'm happy to provide a copy of the slides. We've also come to understand how HIV can be controlled in the absence of medications for some people. And so this began, 20 years ago, understanding elite and viremic controllers, people who could control HIV without medications. Some of those people went a really long time without progressing immunologically, without seeing sort of all of the collateral damage that can happen in a person's body. Over the last 10 years, we've come to understand this rare phenotype of post-treatment control. So how when a very small number of people who are on ART pause their meds, they can have post-ART control for some amount of time. And so there are major efforts to understand that. I think over the last 5 years, we've gotten a better sense of this concept of exceptional control, which is a really extreme end of spontaneous control, where people are able to control their virus without meds for decades with, we don't think, huge immunologic consequences. But there, you can count on one hand how many people we think have achieved this. And then most of what I'll talk about today is this concept of post-intervention control, which is what we're really interested in achieving in the cure space, to try to basically make people who otherwise would not be able to control HIV without meds able to do so by altering their reservoir or their immune system. Next slide. So there are a number of different strategies that are being employed to try to achieve control of HIV in an individual with various interventions. Most of these started by targeting the reservoir, the fundamental problem. And there were strategies that had, cool catchphrases like shock and kill, basically launching out all of the HIV in a person's cells, making it visible, and then wiping out those cells that were occupied by HIV. Strategies like block and lock, which in our group we basically describe as kind of pouring concrete all over the cells where the HIV is so that it can't reemerge. Over the last 10 years, there's been a shift toward focusing on immunotherapy So vaccine approaches to improve T cell responses, cytokines like IL-15, which can alter innate immunity, things like checkpoint inhibitors, which are widely employed in cancer. And I think we're going to see over the next 5 to 10 years a big shift, which I'll talk about, towards cell and gene therapy, actually, giving people's immune systems the ability to better control HIV by fundamentally altering, their immune systems, their cells, and even their reservoir. But I think the consensus among the research community is that it's likely that there's not going to be one silver bullet. We're going to need a combination of interventions from these categories, and even within these categories, to get to a cure. Next slide, please. So the newest kind of catchphrase is reduce and control. And so the concept here is that we need to both reduce the size of a person's reservoir, sort of minimize the amount of HIV that is available in the reservoir to reemerge if people are off of meds, and then optimize their immune system to better be able to better control the virus that is in that reservoir. And the combination of those two things could get us to a cure. Next slide. All right, so I'm going to focus now on some recent progress in immune therapy. So a brief word on vaccines. So there's been a lot of work over the last decade on trying to design better HIV vaccines to alter people's T cell responses, basically, to better target sort of the key epitopes of the virus and, ideally control HIV. There's been some provocative data that has emerged from these studies, but it doesn't seem that vaccines alone, at least the current generation of vaccines that we have, are going to be able to achieve this independent of combinations with other types of therapeutics. And so we have made progress in inducing good responses, but that has not yielded sustained control off of ART. Next slide. What I really want to spend most time on is talking about broadly neutralizing antibodies, which I think have consistently been shown to be a really important tool in our toolbox for HIV cure. This slide shows all of the various types of broadly neutralizing antibodies, or BNABs. These are really, I think about these as monoclonal antibodies that can be manufactured to target the most important areas of the virus to either neutralize that virus itself or to improve immune responses against the virus. Next. This is a schematic that shows all of the various phases of an HIV infection in which broadly neutralizing antibodies have been employed to try to induce control of HIV. So some studies administer broadly neutralizing antibodies on the far left here. As people are beginning their ART treatment, they might receive broadly neutralizing antibodies to try to alter the initial dynamics there. There are also studies in the middle here where we have administered BNABs in people who are on ART and are suppressed with the goal of basically forming immune complexes, making the reservoir more visible, potentially clearing the reservoir. And then many studies have administered BNABs either right at the beginning of the time where participants are pausing their ART, or even as the virus is starting to reemerge. So those are all different strategies that are employed. Mm-hmm. Next slide, please.
I'm running it from out there.
The goal of these strategies is to induce what we refer to as post-intervention control, and if it's after BNABs, post-BNAB control. And what we see across these studies is that not all, but some people exhibit either a delay in their viral rebound. So instead of the virus coming back sort of explosively, as you'd expect at the, in the early days off of ART, it comes back way later or way more slowly than you would expect. In some people, it comes back and then it comes under control, and then it comes back a little bit more and it comes under control. And some people don't rebound at all. And so understanding how and why that happens is critically important to the cure agenda. Next slide. What do we expect in people who are not participating in these immunotherapy trials? Well, we have a pretty good sense of how often people are able to achieve this post-treatment control, and it's not common. So this is a really nice study that came out a couple of weeks ago, looking at the placebo arms from a variety of different prospective trials. They studied about 200 early treated individuals and 200 later treated individuals. And that's important because based upon characteristics of the reservoir or the immune system, that can determine the likelihood that somebody is able to achieve post-treatment control. But the upshot of this is that only about 8% of people achieved low-ish viral loads 3 months after interrupting their meds. That proportion was a little bit higher, almost 14% in the early treated individuals, and was 3% in the later treated individuals. But this is uncommon. That's the point I want to make. Next slide. So what's very interesting is that a number of studies that have employed these broadly neutralizing antibodies have achieved a higher proportion of control compared to what I just showed you. These are small studies. You can see the denominators here, like a dozen people, but they're very, very provocative, and they've, I think, taught us that there's something to do here. Next slide. There are two, go back please. There are two studies that I want to point out that are going to be presented, I think, at CROI this year. One is the Rio study, which studied several dozen people, I think up to 70 people, using a BNAB strategy. And we don't know how many people achieved control in that study, but I think we're going to find out in a couple of days. And then in addition, the Fresh Gilead study, which is done in South Africa, is studying this approach in women living with HIV, most of whom have subtype C virus, which is a really different population than has been studied in all of the other studies on this slide, and is critically important for the global cure agenda. Next slide. I'll spend a minute and talk about our local study at UCSF, which some of you in the room were involved with. So this was the UCSF AMFAR combination study. If you click the button once, this was a 3-stage combination regimen that involved a therapeutic vaccine, so an anti-HIV vaccine meant to improve T-cell responses, followed by infusion of those broadly neutralizing antibodies that I mentioned, with a TLR9 agonist to kind of make the reservoir more visible and form immune complexes. And then after people participated in this with interventions for 34 weeks, they got a final infusion of the antibodies and paused their ART, and we watched to see what would happen. Just to emphasize, the goal of this study was not to replace ART, right? This is not a feasible thing to scale up for millions of people. But it was really to demonstrate that this regimen, which was initially sort of identified in studies of non-human primates, could be feasible and safe in people, and to provide the proof of concept that we need to justify much larger trials with an approach like this. Next. And so what is exciting, I think, about this study is that we identified sustained control after ART was paused in 7 of the 10 participants. That's a lot higher than 14%. 3 of the participants, shown in red on the left, had sort of typical exponential rebound. But if you click one more time, the remainder of the participants achieved set points that were at or around 1,000 copies, which again is not going to replace ART, but is very, very interesting. And so understanding what happened in those individuals is sort of crucial to the future of the field. Next slide. So as I mentioned, in part based on the proof of concept here, this is now being scaled up into other trials, larger trials, randomized trials, to see whether similar results can be achieved. And this is being done by groups like MHRP, by groups like the ACTG, by companies, and by others. Next slide. So how is this achieved? This is a very busy slide, but there are a variety of different mechanisms by which post-intervention control can be achieved, some of which is related to a long-term antiviral effect, some of which may be related to slowing down the activity of HIV as it reemerges to kind of prevent the immune system from falling behind and allow it to keep up. And then some of it might be related to the direct effect that we have in altering a person's immune responses. We don't know which of these responses is most important. We don't know when they are most important. We don't know where they're most important yet. And we don't know if the same responses are important across all comers. So there's a lot of work that remains to be done. Next slide. I think another really important concept in the field of cure right now is that many studies have looked sort of weeks after people have paused their ART after these interventions and tried to figure out what is different between the people who are able to control or not. What we're seeing in our program is that actually the events that are taking place in the very first few days after someone pauses their ART, just as the virus is reemerging and re-encountering the immune system, are probably absolutely critical in figuring out who is destined to control HIV or not. And so I'm not going to spend a lot of time here, but this is some modeling data that is looking at, on the left, typical people who pause their ART and their virus comes back quite exponentially. In the middle are people who are known to be controllers before they were on meds. And they're on meds as part of standard of care because we treat everybody now, but they were willing to pause their meds to see what would happen. And those individuals, the slope of their initial rebound is significantly reduced compared to people who are not controllers. What's really interesting is that if you look at our post-intervention controllers from our immunotherapy study, their initial rebound is even slower than the spontaneous controllers. And so we think that this is starting to move the needle, and we think that understanding immunologically what is happening in these earliest days could really matter. Next slide. And so we're doing a deep dive now, and others are doing similar work to try to understand biologically, immunologically, mechanistically what drives this control. And Rachel Rudeshauser, who's in the back of the room, has made really interesting observations about the sort of expansion and proliferation of the T cell response, even in those earliest days, where that initial response seems to strongly relate to what the ultimate result of control would be. Next slide. Okay, so where do we need to go? We need to do a lot more work to understand the mechanism, and I'm not going to get into all of this, but just to say that we need to understand the role of every single arm of the immune system, which likely play a part in this highly synchronized effort to succeed at controlling HIV, or in this sort of unsynchronized effort that ultimately fails to do so. We need to better optimize our combination therapies. That means making better BNABs. We need to develop new BNABs, and we need to ensure that we have access to these BNABs for clinical trials, which is quite difficult. We need better vaccines, and we need better methods to target the reservoir. Next slide. And then we need to also have the resources to conduct more definitive randomized trials with the optimal timing of biospecimen collection. The initial set of trials that were performed, we were sort of guessing where the interesting immunology would be taking place. And in some cases we captured it, and in other cases we didn't. Now we have way more insight. And so we can more rationally design these studies to get the answers that we need. So this is a really important review that was written a couple of years ago, where Toomey and Mike McCune and Steve called out a number of characteristics of a curative intervention. It needs to be safe, it needs to be affordable, it needs to be scalable, it needs to be effective, and it needs to protect against reinfection. Those are 5 big tasks. We are very unlikely to hit it out of the park sort of on the first try. Yeah.
What does the definition of ART stand for?
Oh, antiretroviral therapy, HIV meds. Yeah. If you advance, so ultimately getting to a cure strategy. I think we can do this, and it's becoming increasingly feasible. But we need to understand that it's going to be iterative. Next slide, please. At a minimum, I think a cure strategy needs to decrease HIV levels below the threshold at which transmission can occur. We typically think of this as 200 copies. There's some data that's emerging that suggests that maybe a slightly higher level is feasible, but I don't really think that that would be good enough, and that certainly would not achieve what we need to do, what we need to achieve for sort of overall individual health. And so, we can debate over what the target threshold should be, but ultimately what we're trying to achieve is something that is as good or better than ART. And it's going to take a lot of iteration to get there. Next slide. So how are we going to get there? So I think a lot of the future is going to be in the realm of cell and gene therapy. And so all of the sort of methods that I've mentioned, reduce, control, combination methods, combination approaches, have sort of related approaches within the realm of cell and gene therapy. And we are thinking more and more about sort of the shift from immunotherapy to these types of therapies, which I'll talk about. So the next slide. The first sort of approach is cell and gene therapy to reduce the reservoir. Okay. This is a trial that is ongoing by Excision, which is a phase 1 and 2 study to actually try to basically edit out HIV in the genome. They're using an adenovector to deliver CRISPR, which you may have heard of, to basically excise HIV from the genome. There's some initial safety and efficacy data that's been reported. It's quite provocative. Seems so far to be safe, and there are sort of reports of some delayed rebound in these cases. So a really interesting approach, just cutting out HIV. Next slide. And then there are also approaches, and I believe that this will be presented at CROI as well, trying to edit CCR5, the receptor on the cell surface that HIV uses. And so in this study called Trailblazer, participants actually underwent a leukapheresis where a huge number of CD4 cells were collected, and then in the lab, edited out were basically the receptor for CCR5, and then the cells were reinfused. So we'll learn more about that this week. Next slide. And then the control element of this. So a lot of the really exciting progress in the last few years has been in the delivery of antibodies, the same types of antibodies that I mentioned, but basically empowering people's cells to make those on their own. And so initially this work was done in the non-human primate model. This is a study that was able to deliver using an adenovirus vector to muscle cells, basically the code that was needed for 2 broadly neutralizing antibodies, which you can see in yellow and green. They achieved high levels of production of those antibodies for years in these monkeys. And in red, you can see that their virus remained suppressed for quite a while. Next slide. This is also being done in adult non-human primates. And so a similar study here, again, delivering the genes that are needed to make these antibodies to macaques with SIV. And they were able to achieve reasonably good levels of these antibodies with limited immune clearance of those antibodies. Next slide. So now these strategies are being translated into people. And so there are a number of studies, a couple of studies now that have shown that this strategy of using an adenovirus vector to deliver the code for these antibodies is safe in people. we're able to achieve measurable levels of the antibodies, but not yet adequate levels. And the main limitation to this is that we can't get a level of expression quite to where we need it to be yet. And we also develop immune responses against those antibodies that clear. Next slide. So there is now a major effort to try to address these challenges. I'm co-leading with Kara Chu and Alex Belez a study within the ACTG that's going to try to optimize delivery of these antibodies. What we're going to do in this study is work with volunteers who will receive a single dose of this vectored antibody, either with 1 shot or 4 shots. It's the same dose regardless, but there's some evidence that actually dividing up the locations where you get it greatly improves the expression. And then we're going to do this either with or without a few weeks of steroid medications to kind of modulate the immune system and minimize those antibody responses. So this study is supposed to launch this fall. This is not going to cure people, but this is going to be a really important sort of first step in understanding how to optimize these approaches. Next slide.
ATI.
There's no ATI in this study. And then you may have heard of the Miami monkey, which is a monkey in Miami which I think actually lives in the Midwest, that has been cured by a similar approach, and now there's actually industry engagement on this topic. So that's great. Next slide. And then in addition to these vectored approaches, there's a major effort around CAR T-cell approaches now. Interestingly, if you look back historically, CAR Ts were initially studied in HIV 20 years ago, have been totally transformative for cancer and are becoming transformative for autoimmune conditions. And now there are studies, including by our group and the group at Penn, trying to translate this back to HIV. Next slide. So a few other words on what we need. We need better diagnostics. One of I can't overstate how committed the participants in these studies are. The amount of laboratory monitoring is insane. It's a huge commitment. Having diagnostic tests that could detect virus that people could administer to themselves at home would be totally transformative. Not enough investment in this area. Next slide. This is the same point, we can skip it. And then we need more socio-behavioral research to know what people with HIV want a cure to look like, what risks are acceptable, and what challenges people face participating in these highly, highly intensive studies. This is just calling out one of our local participants, Clark, who's been hugely committed to our program and has talked about his experiences, and then work by Corrine and John and others, and many in this room. Next slide. So there are a few threats. Industry engagement is too weak. It may be waning. There are some companies that are very involved. Many are less involved or are shifting away, which is quite concerning. We in academia are extremely committed to this, but we don't have the resources to translate treatments from the lab to the clinic. And then there's huge uncertainty, especially now, regarding key programs that are highly, highly intertwined with the cure agenda, as you all know. The Gates Foundation is doing a lot of important work to try to drive this forward. They are, really a crucial player in this, trying to identify strategies that can be tested head-to-head, that can be sort of committed to and scaled up. And I think that their role in the next 5 to 10 years is going to be absolutely critical. Next slide. The Vaccine Research Center at NIAID has been totally crucial to identifying DNABs, identifying treatments, making them available, getting them into the clinic. And I think we all have a lot of concern over whether that will be sustained. Next slide. And then there are these collaboratories at Duke and at Scripps that are hugely important in manufacturing agents that can be used in cure strategies, getting these to the clinic and being able to scale them up. And I think we need expansion of programs like this one. Next slide. So just to wrap up here, and this is my last slide, this is sort of the map of where we need to go to get to a cure. We are at the front end of this map still, but we are making steady progress forward. I think the point I want to make is that we are making progress. The progress is certainly under threat, but there are many people who are highly motivated to continue this work. The first round of cure strategies are not going to replace ART, and they should not be perceived as trying to do that. This is really going to be a highly iterative approach where we learn from each success and we learn from each thing that does not work to kind of get us where we need to be. But ultimately, the goal of this whole agenda is to get to a strategy where basically a single therapeutic, a single shot could cure everybody, regardless of who they are, regardless of where they are, and can protect them from HIV going forward. So I will end there, and thanks so much for your attention, and happy to take questions if there's time. Thank you.
About doable and scalable and all of that. I'm the community representative on A5374 and A5386, which are two cure trials in ECTG. Well, we are putting these participants through, I mean, daily, the emails are coming.
Maybe it should go up and use the microphone.
Daily, the emails are coming from sites,
Just don't pull too hard.
study nurses saying, we have this issue with this participant, or we have the issue with the leukopax, or we have the issue with this, we have that. Should we bring him back in? And the team has to kind of confer behind the scenes sometimes. Then we have to get back with an answer. Thank you so much and all that. And it's not scalable, So what we're trying is, we have to learn from this so that we can maybe come up with something. But that's a huge challenge because what these participants are going through could not happen in real life.
Yeah. I totally agree. I'm not sure I have anything to add. I think that, this is sort of phase one of figuring out what works, what doesn't work, and how to hone the next generation of strategies. Jeff.
Yeah, you mentioned the adenovirus. This is a way to do the one-shot approach, but there's a lot of pre-existing resistance to that, and it could be different in different regions and so forth. And how are they planning to address this so there's not an off-the-shelf thing that doesn't work for a lot of people, and are there going to be backup treatments for those for whom it doesn't work?
Yeah, there are a lot of challenges related to that approach, and we're still a ways away from being able to scale that too. But I think using the current generation of adenovirus vectors to understand how to optimize delivery, and that and then hopefully we'll be able to solve that problem, but it's not easy.
Thank you so much for the great talk. I have two questions. One, could you please throw more light on the statement that there is better understanding of the reservoirs? It's a statement I've been hearing for the last 2 decades, and still we don't understand the reservoirs.
Yeah.
Two, given the current global mess,
Yes.
and confusion, especially in my country, Uganda, Africa, what's wrong with having an intervention which is potent, safe, minus protection? What's wrong with it?
Yeah. So the first question was about the reservoir, and it was pointed out that I said that we've made a lot of progress on the reservoir, but others say that we still don't understand the reservoir, and it is definitely true that both of those things are true. I think every time we think we understand things fully, there's like a new wrench. So it's the size, it's the intactness, it's the replication competence, which is not the same as whether it's intact. It's the location of the reservoir. And so, yeah, I mean, I concede the point. The second question was, would a cure strategy that achieves 4 of the 5 characteristics outlined in that paper, but does not protect against reinfection, be acceptable? And I think that is a valid question that we all sort of need to come up with an answer to together. I mean, if we could achieve something that does 3 or 4 of those 5 criteria, that would still be progress. But I think ultimately, the goal is to eventually hit a home run.
Thank you.
I think we're out of time, but I'll be around if people want to chat. And thanks so much.